So the glucose molecule is at the center of all metabolism that occurs inside of a human cell. So when the glucose molecule enters inside of cells, these glucose molecules go through a pathway referred to as glycolysis. And in glycolysis, we take glucose molecules and we do a bunch of chemical reactions, eventually taking those glucose molecules and converting them into pyruvate molecules. And keep in mind, each of these chemical reactions are catalyzed by their own unique enzymes. So now, once we go through glycolysis and form these pyruvate molecules, then they can enter inside of the mitochondria. And in the mitochondria, we take these pyruvate molecules and convert them into acetyl-CoA molecules. Now, once we form these acetyl-CoA molecules, they can react with oxaloacetate molecules to form citrate molecules. And once we form these citrate molecules, again, we can go through a bunch of chemical reactions, eventually reforming oxaloacetate molecules, which can react with new acetyl-CoA molecules to form new citrate molecules to go through this cycle over again. So this pathway is referred to as the Krebs Cycle. But these two pathways, glycolysis and the Krebs Cycle, are collectively referred to as central metabolism because glycolysis and the Krebs Cycle are at the center of all metabolism that occurs in the cell. And nearly all cells in the human body go through central metabolism. Why? Well, when we take glucose molecules to go through central metabolism, we create ATP molecules. And we know these ATP molecules are high energy molecules, which are used as a source of energy as a source of fuel to energize all the energetic chemical reactions the cell needs to survive. But not just that, when we take glucose molecules to go through central metabolism, we also create these reduced cofactors like NADH and FADH2. And we know these reduced cofactors have electrons that can be donated to the electron transfer chain to create even more ATP. So the point is, when we go through central metabolism, we create the ATP that's needed for the cell to survive. But what's interesting is, we can take glucose molecules to go through central metabolism, but we can also take other monosaccharides to enter central metabolism. For example, galactose. We can take galactose and convert it into this particular intermediate. Now, once we form this particular intermediate, it swoops in to central metabolism and can be used to create ATP. So the point is, any molecule that can be converted into one of these intermediates can simply swoop in and be used to create ATP. And lots of different monosaccharides can be converted into intermediates and be used to create ATP. So what about proteins and their component amino acids? Can we take these amino acids to enter central metabolism to create ATP? Well, it depends, because keep in mind, amino acids have a carboxyl group and an amine group. But notice, none of these intermediates of central metabolism have nitrogen molecules. So therefore, if we want to take amino acids to enter central metabolism, we must get rid of this nitrogen group. But we can do that. We can get rid of this nitrogen group and replace it with the carbonyl group. Once we do that, now we have this carbon backbone, and these carbon backbones can enter central metabolism. And notice alanine, when we get rid of the amine group and replace it with this carbonyl group, we form this molecule, which happens to be pyruvate. So this can simply slip in as pyruvate and enter central metabolism and be used to create ATP. So this is true for any generic amino acid with any R group. And again, it's the same idea. We get rid of the amine group, replace it with the carbonyl group, and now we have this carbon backbone, which we refer to as an alpha keto acid. And we know different amino acids will have different R groups, but depending on what that R group is, we can do chemical modifications to convert these keto acids into intermediates of the central metabolism, and therefore enter central metabolism and be used to create ATP. But you might wonder, when we take these amino acids and get rid of this amine group to form these carbon backbones to enter central metabolism, what do we do with these amine groups? Because we know ammonia is toxic to the cell. So what do we do with all this ammonia? Well, we usually have a nitrogen acceptor, and the universal nitrogen acceptor is alpha ketoglutarate. So this alpha ketoglutarate accepts that nitrogen. So this nitrogen waste we donate to alpha ketoglutarate to form glutamate. Now, depending on what the cell was, there will be this nitrogen transporter where we can take this nitrogen from glutamate, donate it to the nitrogen transporter. Then this nitrogen transporter can enter the blood and transport that nitrogen to the liver hepatocytes, where then that nitrogen is donated to another alpha-ketoglutarate to form glutamate, which can then safely enter the urea cycle, where we take that nitrogen and convert it into the safe urea, which can safely be disposed out of our body. But only the liver can safely dispose of nitrogen. So the point is, if we have a tissue that wants to use proteins and amino acids as a source of energy, we must get rid of that nitrogen group. But how do we get rid of that nitrogen group? Well, again, we donate that nitrogen, which goes to the nitrogen transporter, which can then safely donate that nitrogen to the liver, and the liver can safely dispose of that nitrogen through the urea cycle. So what about fatty acids? Can lipids and fatty acids enter central metabolism and be used to create ATP? Well, it's a little complicating. So if we want to use fatty acids to create ATP, we take the fatty acids and go through a pathway we refer to as beta oxidation. And every time we go through a round of beta oxidation, we create an FADH2 and an NADH, and we lose 2 carbons in the form of acetyl-CoA. So that happens every time we do a round of beta oxidation. So now we have a fatty acid that's 2 carbons shorter. Now, once we form this fatty acid, we can do another round of beta oxidation. And again, every time we do a round of beta oxidation, we create these reduced cofactors and we lose 2 carbons in the form of acetyl-CoA. So we know these reduced cofactors can directly fuel the electron transfer chain to create ATP. So therefore, these fatty acids, the way they are used to make ATP is directly by forming these reduced cofactors to fuel the electron transfer chain. And again, we also create these acetyl-CoA molecules, which theoretically could enter the Krebs cycle, but in reality, it's a little more complicating. So we'll talk about that later. So we've already learned when we take glucose molecules and go in this direction to form pyruvate molecules, that pathway is referred to as glycolysis. However, we can also go in the reverse direction. We can take pyruvate and go in the reverse direction, forming glucose molecules. And we can do this because each of these chemical reactions were reversible. However, once we take pyruvate and form acetyl-CoA, this is irreversible. This is a one-way street, and we cannot go in the reverse direction. Once we form acetyl-CoA, we cannot go in the reverse direction to form pyruvate. However, pyruvate, we can go in the reverse direction to form glucose molecules. So when we go in this reverse direction, it's referred to as gluconeogenesis, the genesis of new glucose molecules, where we go in this reverse direction and we create new glucose molecules. So in certain contexts, we need to go in this reverse direction to form new glucose molecules. However, glucose is made out of 6 carbons. So if we want to make glucose molecules, we need a source of carbons. So what are those major sources of carbons? Well, lactate has 3 carbons. So lactate can be converted into pyruvate, and once we form pyruvate, we know we can go in the reverse direction to form glucose molecules. So therefore, these 3 carbons in lactate can be used to form glucose molecules. The same thing with glycerol. We know we have these triglycerides, which is a storage form of energy. But when we hydrolyze these ester linkages and release a glycerol molecule, this glycerol molecule has 3 carbons. And glycerol can be converted into this intermediate, which we know can then be used to make glucose molecules. However, what about keto acids, like beta-hydroxybutyrate? Can these atoms be used to biosynthesize glucose molecules? Well, alpha keto acids can be converted into acetyl-CoA. But we know once we form acetyl-CoA, we cannot go in this reverse direction to form pyruvate molecules, so we're stuck. Once we form acetyl-CoA, we are forced to go in this direction. So therefore, these atoms in keto acids cannot be used to form glucose molecules. So what about proteins and these amino acids? Can we take these atoms and amino acids to form glucose molecules? Well, it depends. For example, alanine, we know when we get rid of the amine group, we form this alpha keto acid. which happens to be pyruvate. So we know this can enter as pyruvate, which can be used to make glucose. So therefore, these atoms in alanine can go through this pathway to form glucose molecules. What about aspartate? Well, we know we need to get rid of the amine group, so now we form this alpha keto acid. But this particular alpha keto acid, just by chance, happens to be oxaloacetate. And something you just have to memorize is, just by chance, we happen to have an enzyme that can take oxaloacetate to form pyruvate. And once we form pyruvate, we know we can form glucose molecules. So therefore, aspartate can go through this pathway to form this intermediate, which is again this intermediate, which can then be used to form glucose molecules. But what about leucine, the amino acid leucine? Well, again, we get rid of the amine group, so now we form this alpha keto acid. But this alpha keto acid, we have the enzymes to convert it into acetyl-CoA. And we know acetyl-CoA cannot be used to form glucose molecules. We know it cannot go in this reverse direction. It's forced to go in this direction, so it can't be used to form glucose molecules. But you might wonder, can't we take acetyl-CoA to go in this direction to form this guy to then be used to form glucose? Well, no. Acetyl-CoA has theoretically 2 carbons that it could donate to form glucose molecules. However, once acetyl-CoA goes through this pathway, it loses 1 carbon as carbon dioxide, then it loses its 2nd carbon. So therefore, these 2 carbons are lost as carbon dioxide by the time it forms this intermediate. So therefore, these carbons cannot be used to form glucose molecules. But what about methionine? Well, again, we get rid of the amine group, so we form this alpha keto acid. And this particular alpha keto acid can be converted into this particular intermediate, which again can enter this pathway and be used to form glucose molecules. So therefore, certain amino acids can be used to form glucose molecules. And the amino acids that can be used to form glucose molecules are referred to as the glucogenic amino acids. However, other amino acids cannot be used to form glucose molecules, so they're referred to as ketogenic because they can be used to form ketone bodies. So what about fatty acids? Can these atoms in fatty acids be used to create glucose molecules? Well, we know when we go through rounds of beta oxidation, we create acetyl-CoA molecules. And we already explained these carbons in acetyl-CoA cannot be used to form glucose molecules because those carbons are lost as carbon dioxide. However, if you happen to end up with 3 carbons in your final fatty acid, this just by chance can be converted into this intermediate, and it can be used to form glucose molecules. So these last 3 carbons can be used to form glucose molecules. So these 2 carbons in acetyl-CoA are incredibly important. And yeah, even though these 2 carbons cannot be used to form glucose molecules, they can be used for other purposes. For example, we can take these 2 carbons in acetyl-CoA to biosynthesize free fatty acids like palmitic acid through the process of fatty acid synthesis. Now, once we form this palmitic acid, it can react with the moiety of glycerol, where we take 1 moiety of glycerol, 2 moieties of palmitic acid, and 1 inorganic phosphate to form these phospholipids. And we know these phospholipids are key components of our cell membranes. But not just that, we can take 1 moiety of glycerol and 3 moieties of palmitic acid to form triacylglycerides through the process of lipogenesis. And we know these triacylglycerides are the primary storage form of energy our bodies use. For example, when we're in the fed state, so we've just eaten a big meal, we know when we eat a big meal, our bodies will have lots of glucose, proteins, and fatty acids that we got from our meal. So we want to store those molecules. For example, all those glucose molecules we enter through a process of glycogenesis, where we take those glucose molecules and store them in the form of glycogen, which we know is just a storage form of glucose molecules. So we take excess glucose molecules and store them in the form of glycogen. And all those excess proteins and those amino acids we use to replace already existing enzymes and structural proteins and peptide hormones. However, eventually we're going to fill up our glycogen stores, where we filled up our glycogen stores and we've replaced all of the enzymes and structural hormones and proteins. So once we do that, what do we do with all these excess glucose and protein molecules? For example, let's say we've eaten a very big meal. So what do we do with all the excess glucose and proteins? Well, we enter them through central metabolism and use the energy in these molecules to form triacylglycerides. So we convert the excess glucose and proteins into triacylglycerides as a storage form of energy. So we know these triacylglycerides is a primary storage form of energy our bodies use. But also what's pretty interesting is we can take the atoms in serine and react it with these atoms in palmitic acid to form this sphingosine backbone. And we know this sphingosine backbone is very important to form other important lipids. For example, we take this sphingosine backbone and we react a free fatty acid to this amine group, or we add a free fatty acid to this amine group to form ceramide, the ceramide molecule. And we know with the ceramide molecule, we can use it to form lots of important molecules. For example, we take ceramide and we add a phospho group to this hydroxyl head group to form sphingomyelin. And we know the sphingomyelin is a key component of the myelin sheaths of our neurons. Also, what we can do is we can take the ceramide and we can add a carbohydrate to this hydroxyl head group to form cerebrosides. And also, we can add more carbohydrates to form gangliosides. So that's what happens during the fed state. But what about the fasted state? For example, let's say we haven't eaten a meal for a couple days. What is the source of energy if we don't eat? Well, we can use these triacylglycerides as a storage form, as a source of energy, where we hydrolyze these ester linkages, releasing free fatty acids. And we know we can take free fatty acids and go through rounds of beta oxidation. And every time we do a round of beta oxidation, we create reduced cofactors and acetyl-CoA molecules. And we know these reduced cofactors can fuel the electron transfer chain to create ATP. So this is one way we create ATP during a fasted state. We use our triacylglycerides, our storage form of energy, as a source of energy. But we know when we go through rounds of beta oxidation, we also create these acetyl-CoA molecules. So what do we do with these acetyl-CoA molecules? Well, we can react 3 acetyl-CoA molecules with each other to form this HMG-CoA. And this HMG-CoA is a very important molecule because in the liver, in hepatocytes, we can take HMG-CoA and go through a process referred to as ketogenesis, where we form these keto bodies like beta-hydroxybutyrate and acetoacetate. So during a fasted state, with the fasted state hormones and with a lot of acetyl-CoA molecules that we get from beta oxidation, we can form HMG-CoA and go through ketogenesis to form these keto acids in the liver. But why is the liver forming all these keto acids? Well, these keto acids can leave the liver, enter the blood, and enter into other tissues. And these other tissues can take these keto acids and convert them into acetyl-CoA molecules. And then these tissues can use these acetyl-CoA molecules to enter the Krebs cycle to create reduced cofactors to fuel the electron transfer chain to create ATP. So this is what happens during the fasted state. However, this HMG-CoA is really important and can enter other pathways. So again, in a fasted state with the hormones during a fasted state, we take HMG-CoA to form keto acids through ketogenesis. But in other contexts, for example, during a fed state, we can take HMG-CoA and use it to form these 5 carbon units. These 5 carbon units, which can be used as building blocks to build larger organic carbon-based molecules. For example, we can take 2 of these 5 carbon units, react them with each other to form a 10 carbon unit. Then we take a 10 carbon unit, react it with the 5 carbon unit to form a 15 carbon unit. Then we react two of those 15 carbon units to form a 30 carbon unit, which is referred to as squalene. So this squalene has free rotation among some of these bonds, so this squalene can fold and get in a particular conformation where it can react with itself to form lanestrol, which can then be used to form cholesterol. But notice this is pretty interesting. As long as we have glucose molecules, we can take these atoms and glucose molecules to form acetyl-CoA molecules, which can form HMG-CoA, which can form these 5 carbon units, which can be used to form larger organic molecules like cholesterol. So we can biosynthesize cholesterol. And that's good because we know cholesterol is super important and is used to create lots of important hormones. For example, we can take cholesterol, react it with some energy from sunlight, and then reacted with the liver and the kidney to form different derivatives of vitamin D, which is an important hormone. We can also take cholesterol and use it to make lots of other important steroid hormones. For example, in the testes, we can go through this pathway to form testosterone. In the ovaries, we can go through this pathway to form estradiol. In different parts of the adrenal cortex, we can go through different pathways to form cortisol and aldosterone, these important steroid hormones. And in the liver, we can take cholesterol to form these bile salts, which are needed to emulsify and absorb fats from our diet. But what's pretty cool is we can take these intermediates of central metabolism and use them to form amino acids. But we know amino acids have nitrogens, and none of these intermediates of central metabolism have nitrogens. So therefore, to take these intermediates to form amino acids, we need a source of nitrogen. And that nitrogen usually comes from glutamate, which is a universal nitrogen donor. So what are some examples? Well, we can take oxaloacetate, which is an intermediate of central metabolism. So we can take this oxaloacetate and use it to form aspartate. And we can do that because glutamate has a nitrogen that it donates to oxaloacetate to form aspartate. So that's pretty cool. We took this intermediate of central metabolism and formed aspartate. Another example is we can take this particular intermediate of central metabolism and form alanine. And again, we know we were able to do that because we have glutamate, which can donate nitrogens to these intermediates of central metabolism to form certain amino acids. So that's pretty cool. We can take these intermediates of central metabolism to biosynthesize certain amino acids. And here's a comprehensive list of all the amino acids we can form. For example, this intermediate of central metabolism can be used to form serine, which can then be used to form cysteine and glycine. This intermediate can be used to form aspartate, which can be used to form asparagine. And also, all these amino acids with these red stars, we make with very low efficiency. We take this intermediate and form phenylalanine with very low efficiency. So therefore, most of these essential amino acids we need to get from our diet. But a lot of amino acids we can biosynthesize, which is pretty neat. And we know these amino acids are super important to form proteins, to form enzymes and hormones and transcription factors and etc. So we already explained how we can take this intermediate of glycolysis to form serine. But this serine amino acid is really important because it has this carbon, which is used as a carbon donor to build larger molecules. So how do we do this? Well, we take this carbon and we donate it to tetrahydrofolate to form this methylated form of tetrahydrofolate. Then we can take this carbon to donate it to homocysteine. So now homocysteine gains that carbon to form methionine. Now methionine reacts with ATP to form this S-adenosyl methionine, this SAM molecule, which is the universal methyl donor in the body. So again, if we want to build molecules, we need carbons. So a lot of the times those carbons come from SAM. And also if we want to methylate DNA, those carbons come from SAM. So this SAM is the universal methyl donor where it has these carbon groups that it can donate for anabolism. But also keep in mind, this tetrahydrofolate also has a carbon that can donate for anabolism. So these are the two universal carbon donors, the two universal methyl donors. What's also pretty neat is we can take certain amino acids and other sulfur-containing compounds from our diet and use them to form sulfate. Once we form sulfate, we can react it with ATP using this enzyme to form this particular intermediate. Then we can react it with another ATP molecule using this enzyme to form this 3-phosphoadenosine 5-phosphosulfate, this PAPS molecule, which is the universal sulfur donor. So if we need to build molecules that need sulfur molecules, they usually come from this universal sulfur donor. So for example, we know there are lots of different monosaccharides our bodies use for lots of different purposes. For example, we sometimes glycosylate lipids. So we need different monosaccharides to glycosylate these lipids. Also, proteins get glycosylated. So these proteins get glycosylated with lots of different monosaccharides. And we also know red blood cells have proteins that get glycosylated, and that's the basis of blood type. So we know there are lots of different monosaccharides that our bodies use for lots of different purposes. But how do we create these monosaccharides? Well, usually they come from these intermediates of glycolysis. And if you're interested, here are the pathways. I know this looks really complicated, but the point is we can take this intermediate of glycolysis to go through this pathway to form all of these monosaccharides. And again, notice this monosaccharide has a sulfur. Where does it get that sulfur? Well, again, we already explained PAPS is a universal sulfur donor. So once we have this intermediate, we take this PAPS to donate a sulfur to form this sulfated glucosamine. And also we can take this intermediate of central metabolism and throw on a nitrogen from glutamine to form this monosaccharide with the nitrogen group, which can then enter different pathways to form lots of different monosaccharides. And again, whenever a monosaccharide has a sulfur, you know, got it from PAPS. So I know this looks complicated, but the point is there are lots of different monosaccharides we can biosynthesize, and usually they come from these intermediates of central metabolism. We can also take this particular intermediate of glycolysis to enter a pathway referred to as the pentose phosphate pathway. And in this pentose phosphate pathway, we produce two important products. One, we create ribose 5-phosphate, and also we create these reduced NADPH cofactors. And these reduced NADPH cofactors are really important because they have electrons. And essentially, they can use these electrons and these hydrides as a source of electrons for anabolism. Because we know if we want to build larger molecules, we need electrons to form these bonds. So these electrons usually come from NADPH, which has electrons. But also, these NADPH can take their electrons and donate them to this oxidized glutathione to form reduced glutathione. And this reduced glutathione is an endogenous antioxidant. This is an antioxidant. So if we have reactive oxygen species and if we have free radicals, we know these are toxic molecules. They're super reactive because they have these unpaired electrons. So they're toxic. They react with biomolecules and destroy them. So these reactive oxygen species and free radicals are toxic. However, fortunately, we have this endogenous antioxidant, this glutathione, to donate electrons. So now this unpaired electrons gets paired to form a neutralized safe molecule. So that's pretty neat. Essentially what we can do is we can take these electrons from glucose, go through this pathway, take those electrons, and eventually donate them to NADPH, which can donate those electrons to glutathione to form an antioxidant, which can then take those electrons to donate them to free radicals to neutralize those free radicals. So we have this endogenous antioxidant system, which is pretty neat. And we know this is important because our electron transport chain in our mitochondria is constantly producing free radicals. Even if you're healthy, young, whatever, our mitochondria are constantly producing free radicals, which get metabolized into other toxic free radicals. And these free radicals, for example, hydroxyl radical, is extremely dangerous and toxic. It can react with free fatty acids, which react with these hydroxyl radicals to form this peroxy radical, which goes through a very nasty free radical chain reaction, which forms more free radicals and can form carcinogens, and this is a very toxic pathway. However, fortunately, we have this reduced glutathione, which got its electrons from glucose. So this reduced glutathione is an antioxidant. It can donate electrons to vitamin C, which can then donate that electron to vitamin E, which can then donate that electron to this peroxy radical, stopping that toxic chain reaction. So that's pretty neat. We have this endogenous antioxidant system. So it's important to have a strong endogenous antioxidant system by having lots of vitamin C and vitamin E, and also having the right minerals that's needed for these enzymes in this pathway. For example, selenium and magnesium and copper and zinc. These are important minerals to allow a very strong endogenous antioxidant system. However, what about this ribose 5-phosphate? Well, we can take this ribose 5-phosphate and we can add a nitrogenous group to form this nucleic acid precursor, where these atoms came from aspartate and these atoms came from glutamate to form this nucleic acid precursor, which can be used to form pyrimidines like cytidine and uridine, these nucleic acids. So that's pretty cool. And another example is we can take this ribose 5-phosphate, add a different nitrogenous group to form this nucleic acid precursor, where these atoms came from glycine, these atoms came from glutamate, and these atoms came from aspartate. And then these 2 carbons came from tetrahydrofolate, which is a methyl donor. But then we form this intermediate, which we know can be used to form purine nucleic acids like adenosine and guanosine. So that's pretty cool. We can biosynthesize these nucleic acids. As long as we have glucose to go through this pathway to form ribose 5-phosphate, which makes this component, and as long as we can form these amino acids to donate the atoms for this nitrogenous base, we have everything we need from central metabolism to biosynthesize nucleic acids. So that's pretty cool. But also we can degrade these nucleic acids. For example, GTP, we can degrade it where we get rid of the phosphate groups, then we hydrolyze this bond, releasing this nitrogenous base, which we can then catabolize to form uric acid, which can safely be disposed in our urine. And the same thing with the pyrimidines. We can degrade these pyrimidines to form these end products, which can, for example, CO2 we can simply breathe out. Ammonia gets converted into urea, which can be safely exposed of, and these compounds can also be safely exposed of. So we can biosynthesize these nucleic acids, but we can also safely degrade them and get rid of these waste products. We can also use these intermediates of central metabolism to biosynthesize neurotransmitters. For example, we can take this intermediate to form phenylalanine. And the truth is we do this with a very low efficiency, so most of the phenylalanine we need from our diet. But once we have phenylalanine, we can form tyrosine, which can be used to form L-dopa, which can be used to form dopamine. So that's how we biosynthesize dopamine. But we can also go a step further if we need norepinephrine. And we can also go another step further if we need epinephrine. So this is how we biosynthesize a lot of neurotransmitters. There are other pathways. For example, we take acetyl-CoA and react it with this choline molecule to form acetylcholine, which is a really important neurotransmitter. We take this particular intermediate to form glutamate, which can be used to form GABA and glutamine. These are 2 important neurotransmitters. We can form tryptophan with very low efficiency, so most of the tryptophan we need from our diet, but once we have tryptophan, we can then form serotonin, or go a step further to form melatonin. And we can also take histidine to form histamine. So that's how we biosynthesize a lot of these important neurotransmitters, but a lot of them come from these intermediates of central metabolism. Also, we can take this intermediate to eventually form glycine, which we can react with succinyl-CoA to form this particular intermediate. Then we take 2 of these guys and react them with each other to form this compound. Then we take 4 of these guys, react them with each other to form this porphyrin ring. And we know this porphyrin ring with this iron group can be used, this porphyrin ring can be used to form hemoglobin or myoglobin or the cytochromes important for metabolism or the electron transfer chain. So that's pretty cool. We can form this porphyrin ring from these intermediates of central metabolism. So now you can see why glycolysis and the Krebs cycle are collectively referred to as central metabolism, because these 2 pathways are at the center of all metabolism that occurs in the cell. All these different metabolic pathways stem from these intermediates of central metabolism. And if you're interested, in the link below, I have a different link for each of these pathways in more detail. For example, I have a separate link dedicated for amino acid biosynthesis, and another link dedicated to ketogenesis, and another link dedicated to fatty acid synthesis. I have different links dedicated to each of these chemical reactions. So one last final note. Yeah, we can take these intermediates of central metabolism to biosynthesize lots of important compounds. However, there are limitations to central metabolism. For example, by definition, we cannot biosynthesize vitamins. We cannot take glucose and these intermediates to biosynthesize vitamins. For example, vitamin B3 niacin. Yeah, this may be a very simple molecule. However, we simply don't have the enzymes to take these intermediates to biosynthesize niacin. So therefore, this vitamin we need to get from our diet. So that's true for all vitamins. We, by definition, cannot biosynthesize them. So these vitamins we must get from our diet. And the same thing with minerals, with electrolytes. For example, sodium and potassium and chromium. We cannot take these intermediates. We can't take glucose with these carbons and oxygens and hydrogens and transmutate these elements to form sodium ions or potassium ions. So therefore, these minerals we must get from our diet. So all these elements in purple are the minerals we must get from our diet. And these minerals are really important. Some of these minerals, for example, chromium is really important for certain enzymes to function properly. So there are limitations to what we can do with central metabolism.